Hello, and thank you for joining today's cutting edge conversation about the role of in vivo studies in personalized medicine. We appreciate your involvement. We will begin by introducing the speakers, then we will get into the content of the webinar and wrap up with the live question and answer portion. If you have any questions at any point please ask them by using the Q&A function, which you should see at the bottom of your Zoom window with the large letters Q&A. We will answer any submitted questions at the If you're interested in getting in touch with any of the speakers after the webinar, their contact information will be available on the last slide. Now we'll begin with introductions. My name is Megan Loy, and I'm the Category Director for In Vivo Services at Scientist.com, as well as a board member of the North American 3Rs Collaborative. For the last 2.5 years, I've been working closely with our clients and suppliers to advance relevant categories, as well as offer animal welfare quality and compliance support. I've been working in the e-marketplaces space for the past 4 years, and previously I spent the majority of my career at Covance Laboratories in Madison, Wisconsin, working both in the vivarium and in client services. I have a bachelor's degree in agricultural sciences and a master's in biotechnology. Today we are joined by Jonathan Nakashima. Jonathan is the Chief Scientific Officer at Certis Oncology. He has more than 15 years of experience developing preclinical oncology models. Under a California Institute for Regenerative Medicine Fellowship, he received his doctorate in medical and molecular pharmacology at UCLA, where he developed genetically engineered and patient-derived xenograft models to study cancers of the central and peripheral nervous systems. He continued on with his postdoctoral training in the Department of Neurobiology, where he received a Kirstein National Research Service Award to study the cell interactions in the brain tumor microenvironment. After his academic training, he joined Crown Biosciences, where he served as a study director for in vitro and in vivo projects, including solid tumor PDX studies, PDX screens, and syngeneic studies. He joined Certis in 2018 as employee number 3 and oversees the company's R&D initiatives, patient-derived orthotopic PDX, OPDX, pharmacology studies, and the development and implementation of translational oncology assays for drug development. Our other speaker today is Dr. Grace Walton. Grace earned her PhD in nutrition sciences at the University of Alabama at Birmingham, where she studied NR4A nuclear receptors in the context of diabetes and insulin resistance. She moved to the University of Kentucky for a postdoctoral fellowship, where she studied molecular mechanisms of exercise physiology. She then remained at Kentucky as a staff scientist and then as a research assistant professor, where she continued to study exercise physiology in vitro, in mice, and in clinical trials. She has published numerous first author papers in high quality journals and is excited to pivot to oncology work at Hera BioLabs. Thank you both for joining me today. I will now begin with the introductions of the current challenges surrounding personalized medicine and in vivo studies. Personalized medicine, also known as precision medicine or individualized medicine, is prevention or treatment of a disease that is tailored or customized to the unique characteristics of the patient. Going beyond classic factors, personalized medicine takes advantage of the patient's genetic profile and moves us away from the one-size-fits-all approach. On the upper half of the slide, you can see the traditional approach to treatment. These 4 patients all receive the same treatment, but because they're all different genetically, they have different responses. Maybe 2 react well to treatment, 1 man has no effect from the drug, and 1 woman actually has an adverse reaction. The lower half of the slide represents an example of a personalized approach. The patients undergo blood and tissue analysis, and the therapies are tailored to those results. Some may get the common treatment that the traditional patients received, but the others receive different treatments. And because of this, more or all of the patients respond well. Personalized medicine is not a new concept, but the tremendous progress in the last few decades can make it seem that way. Starting with the identification of the ABO blood groups and the discovery of its impact on successful transfusions in the early 1900s, the recognition of patient biology as a critical player in treatment outcome grew through the latter half of the 20th century. The structure of DNA was laid out in the 1950s, which led to the first DNA sequencing method in the 1970s. Today, having completed the mapping of the human genome, we can identify quite a few genetic markers of disease. While most human disease is created by a complex interaction of genetics and environment, we can at least craft more accurate predictions and apply more impactful treatments. So why don't all patients have bespoke treatment options? There are actually quite a few challenges, which we'll break out into 2 areas. The first is diagnostics. The personalized medicine process generally starts with next-gen sequencing for the patients, which must have a high degree of accuracy if it will be used for diagnostics in future health decisions. The FDA is currently working to ensure these tests and the large amount of data they produce are accurate, although it is posing some novel regulatory challenges. The second area is the actual treatment, whether that is a drug, biologic, or other type of intervention. For a treatment to receive approval, it must be proven both safe and efficacious. But how can you do this with a personalized treatment? First, we must understand that personalized medicine does not mean unique medicine. In this context, personalized refers to subpopulations or groups of people. A doctor may be able to look at a profile and genetic sequence or biomarkers and suggest a drug based on that information, but that doesn't mean the drug won't work for anyone else in the world. And with this lens on, we can see that it does allow for traditional preclinical work. Efficacy, however, is a different story, and we see this issue clearly in oncology. In essence, our challenge is proving that a treatment targeting a specific human disease or tumor is efficacious when treating an animal in vivo. So how do we hit that bullseye? I will let our speakers show you, and we'll start with Dr. Jonathan Nakashima from Certis. The floor is yours, Jonathan.
Thank you. Thank you, Megan, for that introduction, and to the sciences team for having me here. Today, I'm going to talk about how we at Certis are advancing precision oncology and as well as translational drug development through orthotopic PDX models. My next slide is going to just show a little bit of background. Again, Megan covered it mostly, but I think what's important to realize that over the past 20 years, the number of drugs been approved for cancer has dramatically increased, and there are thousands of more in the pipeline. So with the thought of precision oncology, one of the challenges is what drugs with all these new ones available do you choose for? And as Megan mentioned on my next slide, there are a number of different types of precision oncology platforms. First, you have your sequencing, as we mentioned, but then there's more functional tests, and that's like an in vitro screen where you can take the patient's cancer cells in a dish and screen it through compounds. And what we believe is the highest level of precision is the in vivo testing. That is testing a patient's tumor growing in a mouse, so you get the full effects of the biology. But on top of that, we take it one step further in that we do orthotopic implantation. Now, why do we do orthotopic implantation? The main reason is because of the tumor microenvironment. Most scientists know that the tumor microenvironment plays a critical role in tumor biology. And therefore, implanting a tumor ectopically in the wrong location could affect the biology, the response to drugs. And most notably, what we've noticed is that when PDX tumors are implanted orthotopically, they have a higher propensity to metastasize. And metastasis is really what kills the patient, not so much the primary. So we believe that the orthotopic PDX model is the highest level of precision oncology that you can attain. The next slide shows our process here for patients. It's a simple process with many complicated steps in between. The first one is initiation. We'll take a tumor biopsy, and this can be from surgical down to core needle. And right now we're working on SIEs and pleural effusion fluids from patients. We implant those into seed animals to see if the tumor will take or not. If the tumor does take, then we take that, we serially passage it into a number of animals for the cohorts. And then as those tumors grow in that secondary cohort, we randomize and then treat with the drugs that are chosen by the oncologist. Being orthotopic, it's difficult and challenging to monitor drug response. So we have a mouse scale MRI that allows us to monitor tumor volumes over time. The study typically lasts about 4 weeks. And then afterwards, the patient gets a report and they get updates along the way. The next slide is an example of a patient pharmacology study we did. And in this case, it was a pediatric alveolar rhabdomyosarcoma. Now, interestingly, this tumor had a signature genetic translocation. That's a PAX3 FOXO1 translocation. This patient had her tumor sequenced, and given the diagnosis, the oncologist, we already kind of knew that translocation was there. And unfortunately, that translocation is not druggable. So as far as precision oncology with respect to genetic sequencing has taken the field of precision medicine, in this particular case, one can realize that that information does not translate to actions that the oncologist can take. One, we already kind of knew it had that translocation. Two, it confirmed it did, but it did not give any ideas on what to test for this patient. So that's where Certis stepped in. We created a PDX model for this patient. And then we did a large study of about 10 different drugs or 10 different combinations to treat the model, and then give those results back to the patient. The summary of the results can be seen on the left-hand side here through this chart. The drugs range from a number of chemotherapies, combinations of chemotherapies, some small molecule inhibitors, and combinations of SMIs plus chemos as well. Representative images as well as representative drugs are pictured on the left-hand side. You can see that the control grew as expected, but most notably the combinations, the bottom three, either gemcitabine, docetaxel, cyclophosphamide, vinorelbine, and the combination or the novel combination of an HDAC inhibitor, mositinostat, and vinorelbine were the most efficacious combinations. This patient went on to receive cyclophosphamide and vinorelbine. It's considered a maintenance therapy for this disease. And as of today, she still has no evidence of disease after this treatment. So the model proved that this combination was a good combination to choose. But more importantly, what we were able to do in the bottom right-hand corner is take this model one step further. We had great response with about 6 drugs, 6 combinations. And at that point, if we would have stopped and the oncologist didn't know what to choose, it would still be difficult to figure out what one of 6 to put the patient on. So what we elected to do was stop treatment and see what tumors grew back and at what rate they did. And as you can see, the tumors regrew at different rates based off of the therapies that they had received. Most notably, gemcitabine docetaxel had the slowest regrowth rate. So armed with this information, the oncologist now has a secondary play, other options to choose should the patient recur and need a change of therapies. The oncologist has an idea of what to choose now. They can straightly go to gemcitabine and docetaxel or switch to the HDAC inhibitor. That is up to the oncologist's decision, but we have given her medical team some options to pursue should they need there, as well as we have a model that we can work on, and that we've also been on the side developing some resistant models to this. The next slide is just going to demonstrate more reasons why we choose orthotopic. As I mentioned, it recapitulates the tumor microenvironment. And as you can see in the middle panel, it has a high propensity for metastasis. So drug developers who are developing drugs for metastasis or looking at a different type of endpoint in their studies can use orthotopic models to a greater degree than subcutaneous, if you will. And it doesn't show up here due to the transition of my deck to the scientist deck, but on the right-hand panel is a list of and examples of all of our orthotopic implants, ranging from the liver up in the upper left-hand picture, all the way down to the brain, such as glioblastoma in the bottom middle as well. The next slide is just to show that we're not the only ones investigating this. I think the main question is that's great. You have all these models that can metastasize, recapitulate the tumor microenvironment, but does it matter in my study, in my pharmacology study? Here are 3 examples that's been published to show that there are differences in subcutaneous and orthotopic, in particular in response to drugs. And on the next slide, we show here, in this case of a colorectal PDX model that came from a patient's liver, that when implanted either subcutaneously, as in the left-hand side, or orthotopically on the right-hand side, you get a different response based off of the drugs that are applied to this model. Additionally, we show through histology that there's differences in morphology, as well as differences in response, morphological response in this model. We went on the next slide to do gene expression analysis. We show that in this model, there are about 348 genes differentially expressed, and they're all grouped by these categories here. It's a little difficult to see, but the main ones being G protein coupled receptor signaling, immune response, differentiation, all genes that are highly involved in drug response. Later on, I'll show in the case of IO. The next slide here is just another recapitulation across other models, sarcoma, gastric, and pancreatic, showing that when you implant them orthotopically and subcutaneously, you do get differential gene expression across them. And then when we kind of group them together as a meta, you see very similar patterns, differences in immune response, difference in signaling as well. As I alluded to, the next slide here is going to talk a little about in the case of immuno-oncology. So IO is a booming market right now. Everyone is testing new checkpoint inhibitors. What's interesting is that we're not the first ones to show this, but when a tumor is placed in the mouse in different site locations, the investigator will get a different cellular response to that as well. So we went on to do this in the next slide. We did a PBMC engrafted model. Now this is mismatched. It's not the CD34 humanized model. This is simply a PBMC model with a liposarcoma that's high expressing in PD-L1. We implanted it subcutaneously and orthotopically, and we showed that the tumor grows relatively similar. But when you excise the tumor at 1,000 millimeters cubed and you compare the number of T cells with them, you get a different response. There are more T cells in the orthotopic tumor than the subcutaneous. Interestingly, when you look at the type of T cells that are in these tumors, we see that even though the T cells that are less in the subcutaneous, they are more cytoreactive and expressing interferon gamma than the orthotopic tumors as well. So not only are you getting a difference in the number of cells, but you're getting a difference in the phenotype of the cells simply based on where that tumor was implanted. The next slide here shows how these models are how they respond when they're treated with an anti-PD-1 inhibitor. Now, there wasn't a great difference in TGI between subcutaneous and orthotopic, and this could be to the mismatched. There's no myeloid cells. We're well aware of the limitations in the PBMC model. But what we really just wanted to prove out here is that do you get a different response in the same tumor, whether it's implanted subcutaneous or orthotopic, or different sites within the mouse? And the answer is trending towards yes. Again, not a great difference in TGI, but if you look at the number of percent availability of the PD-1, you can see that you're getting much more efficacy, or I should say PD-1 pharmacodynamics in the subcutaneous model than you are the orthotopic model. Furthermore, you get an increase of interferon gamma granzyme B positive CD8 cells in the subcutaneous than you do in the orthotopic. So this is suggesting that when tumors, in case this tumor implanted subcutaneously, the investigator might get a false positive result, and could speak to why 95% of all drugs fail in the clinic. We all know that the models aren't perfect. Perhaps going orthotopically might help push and increase that success rate clinically. Next slide is just to some ongoing questions that we have. Again, this was not HLA matched. This was implanted in the peritoneum of the animal because it was similar to the patient. Question is do these change with the different sites? Does it change with lung, brain, colon, etc.? And then how does the myeloid compartment affect and contribute? And we have some ongoing studies right now in CD34 humanized mice comparing how the cellular response and the efficacy response change or differ between subcutaneous and orthotopic PDX implants. Patients also go through radiation therapy, and many of the times we do these studies isolated in a mouse without recapitulating kind of the full extent of what a patient goes through. So we are also investigating the effects of radiation with our precision irradiator to see how that will also affect the myeloid and lymphoid compartments within a cell. And finally, can we take this one step further, and that's create an autologous humanized model? So on the next slide, I'll show you just some brief flow cytometry staining of our pursuit to create autologous humanized models. So what we're doing now is for patients where we do have enough material, we can also isolate and culture the tumor infiltrating lymphocytes from them. And here's just a facts breakdown of how we analyze them as well. The next slide is just a representative of 3 patients who we've isolated TILs from and created a PDX model from them. Again, you get a different layout or different population distribution based on the patient. So this adds one more level of complexity and speaks to the precision oncology as well. We currently have a bank of about, I believe, 8, and we're trying to build upon that. We have not yet gone into the mouse yet. We have simply created this library of TILs, and we're developing strategies to apply them in the future. Last 2 slides here is just a brief overview of our tumor bank for those who are in drug development. Again, apologies for the small font, but we have a bank of about 100 PDX models right now. We're actively adding more to them, and we expect to be right around about hopefully around 200 by the end of the year, as a number of them are in development as well. If you have any questions, you can ask at the end of the presentation, or on my next slide is the contact information, either to myself or for our business development team. So thank you for your time, and thank you to the scientist team for having me.
Thanks, Jonathan. Next, thank you to everyone that has submitted relevant questions so far. We really appreciate it. And we'll continue on to our next speaker, Dr. Walton, and then we'll go to ask the questions. So go ahead, Grace.
Hi, everybody. I'm Grace Walton from Hera BioLabs, and I'm happy to be here today. First, I'd like to thank the Cutting Edge Conversation staff from Scientist.com for organizing the Precision Oncology series. And today I'm going to tell you how our SRG OncoRat can advance your precision oncology work. Next slide, please. First, I'd like to introduce Hera BioLabs. We offer a suite of proprietary gene editing tools, including the piggyback transposon system and the Cas-CLOVER and Tal-CLOVER gene editing systems. And we provide these as reagents, and we also offer molecular biology services, including vector design, cell line engineering, and animal model generation. On the right-hand side of the slide, our in vivo work is anchored by our SRG OncoRat, or SRG rat. This is a highly immunocompromised rat that is a superior xenograft host. And we're currently using this rat to develop other models, including one with a humanized liver and a humanized immune system. We also offer a number of in vivo contract research services, including oncology studies in our SRG rat, other rats, and mice. And we provide animal model characterization and validation. So let me tell you a little bit more about the SRG rat. The SRG rat was developed on the Sprague Dolly background. It's a double knockout for IL-2 receptor gamma and RAG2. It's a SCID rat. However, it has enhanced immunodeficiency because it lacks B cells and circulating NK cells as well as T cells. In this rat, we get enhanced engraftment rates for PDXs and CDXs, as well as enhanced tumor growth. And then the nice thing about this rat compared to mice is that since it supports larger tumors in larger animals, we can perform serial tumor biopsies, serial blood draws, we can obtain more tissue at necropsy, and that allows us to combine pharmacokinetic, biomarker, tox, and drug efficacy studies. So here's an example of some nice data that we've obtained from the SRG OncoRat. The VCAP line is a prostate cancer line, and prostate cancers are notoriously difficult to grow in commercially available immunodeficient mice. The data on the upper left were obtained following subcutaneous inoculation of the same number of VCAP cells into the hind flank of SRG rats or NSG mice. As you can see, the VCAP growth is far more robust in the SRG rat than it is in the mice. And we often obtain roughly 10 times the tumor volume from SRG rats compared to NSG mice. And this is nice because we don't have to passage as often for PDXs or for cell lines. We can proceed to efficacy studies faster than we would in mice. And the SRG rats also offer the advantage for precision medicine that they accelerate the timeline from a patient biopsy to efficacy testing in animals. So back to the case of the VCAP tumors here, you can see in the lower left that the VCAP tumor in the SRG rat maintains androgen receptor expression and prostate-specific antigen expression after passaging. And then in the upper right, shows an example of how we're able to leverage serial blood draws. So these animals, as the tumors were growing, had blood draws weekly, and we measured PSA in the serum. And then we were able to see this nice tight correlation between serum PSA and tumor volumes as the tumors were growing. And then in the lower right, we show that the VCAP tumor is responsive to standard of care enzalutamide. Next slide, please. So now I'm going to pivot and show you a case study that's part of our ongoing effort to establish new PDX models. In this case, tumor tissue was taken from a patient biopsy and used to establish a PDX in the SRG rat, and we were able to provide actionable data back to the clinic to inform treatment decisions. So, working with our collaborators at the University of Kentucky Markey Cancer Center, we obtained a sample of non-small cell lung carcinoma biopsy from a middle-aged white male, and we used this to develop PDX 3010. We first inoculated the same amount of tissue, 8 cubic millimeters, into NSG mice or SRG rats, and you can see that the time to establishment and the tumor growth was much faster in the SRG rats. And then it's worth noting that from P0 onwards, next slide, please. From P0 onwards, we are able to obtain enough tissue to perform gene sequencing and histology. And in this case, we did histology on tumors taken from the rats at each passage, and we show that they remain positive for TTF1 and negative for P40, indicating that they are indeed the adenocarcinoma and not squamous cell carcinoma. So after establishing tumor in the SRG rats, we then passaged back into NSG mice for efficacy studies. We proceeded to test standard of care treatment against PDX 3010 with cisplatin and docetaxel, and we only observed a moderate response. Then the bar chart on the right-hand side shows percent change in tumor volume in individual mice after starting cisplatin and docetaxel on day 0. Since we didn't see a sufficient response to cisplatin and docetaxel, we proceeded to perform genetic characterization of the tumors in an effort to identify efficacious treatment strategies. So our collaborators in Gautam Narla's lab at the University of Michigan performed targeted exome sequencing and identified a mutation in the MET gene, which was a tyrosine to histidine substitution at amino acid 1248. And this was interesting because substitutions at this tyrosine have been reported in papillary renal carcinomas and are believed to be likely pathogenic. So because we thought that the MET mutation was pathogenic, we wanted to know whether MET inhibitors would inhibit PDX3010 tumor growth. And Dr. Narla found a 2017 publication that showed that the 1248 tyrosine mutation in non-small cell lung cancer did in fact lead to resistance of type 1 MET inhibitors, such as crizotinib. But these same tumors were sensitive to the type 2 MET inhibitor, cabozantinib. So based on these data, Dr. Narla and we hypothesized that PDX3010 would be sensitive to the type 2 inhibitor, cabozantinib. Back in the mice again, in the NSG mice, we proceeded to test the two drugs head-to-head. After dosing for 28 days, we saw no effect of crizotinib, but we saw nearly complete tumor regression with cabozantinib. And then, as with the previous cisplatin study, the histogram on the right-hand side shows percent change in tumor volume in individual mice. Okay, so then to verify the molecular mechanism of tumor regression following cabozantinib treatment, we performed Western blots for phospho and total AKT and phospho and total ERK on the left-hand side following either crizotinib or cabozantinib treatment. And as you can see across the top, the crizotinib failed to really completely suppress phospho-AKT and phospho-ERK, whereas across the bottom left, the cabozantinib completely obliterated the phospho signal. And then on the right-hand side, the IHC underscores the Western blot findings, and you can see that crizotinib does not suppress phospho-AKT and phospho-ERK, while cabozantinib makes them almost undetectable. So in conclusion, the SRG OncoRats are a fantastic host for establishing PDXs, and they enable fast tumor growth, and therefore they shorten the timeline to actually be able to provide data back to clinicians. We've had a lot of success with difficult cancers that are hard to grow in mice, such as prostate cancers. And in the case of PDX3010, we were able to identify a pathogenic mutation and then communicate that back to the clinical oncologist at the Markey Cancer Center. And then we are continuing to work on how the SRG rat can be used as a patient avatar to further enable precision medicine. And so I'd like to thank our collaborators. Dr. Gautam Narla is the Chief of Genetic Medicine at the University of Michigan, and his group did a lot of the molecular biology work on PDX3010. Mark Evers is the director of the University of Kentucky Markey Cancer Center, and he works with us to obtain patient biopsies. And then Fallon Noto is our scientific director here at Hera BioLabs, and she's in charge of R&D and in vivo service. Then I'd like to remind everybody that our products are available through scientist.com. You can obtain SRG OncoRats if you'd like to just purchase rats, or we can design studies for you that we carry out using the SRG OncoRats. We have PDX lines established from lung and ovarian cancers, or we can help you establish your own PDX. We do custom gene editing, including cell line engineering and animal model development. So thanks a lot, you guys, and we're open for questions soon, I think. Have a great day.
Thank you, Grace. That concludes the presentation portion of the event, and thank you to the audience for following along and learning more about the advancements of today and the goals of tomorrow. We are glad that we were able to connect today around the challenges and opportunities available in the in vivo studies and personalized medicine space. We can now get to some of the questions that you all have asked, and please feel free to continue entering your questions into the Q&A section, and I will get to as many as we have time for. So the first question is actually for Jonathan. Jonathan, this attendee did not see any orthotopic models for breast in your slides, and can you comment on that?
Yes, we do have a small bank right now of about 4 breast PDX models. I just didn't show it because you know, they're pretty in the mammary fat pad. You don't really need MRI to do that, and it kind of, the slide was getting a little busy for that way. We have 4, but we have about almost 30 in development right now, in various stages of development. Some are implanted, some are still being acquired that way. But yeah, about so we will have a nice robust breast bank, probably launching in 2022.
Excellent, thank you.
But we do have 4 right now if you're interested in them.
Okay, the next question is for both of you. Personalized medicine is increasingly moving towards proteomics and activated phosphorylated proteomics. Are you both available or aware of this shift in vivo looking at cell signaling?
Yes, I'll go first since I'm already off mute. We have, Certis has a bioinformatics team, and one thing that they're doing right now is developing a gene set enrichment analysis pipeline. So that'll help looking at cell signaling in the vivo models, as well as some AI machine learning driven algorithms to help narrow down the choices, and based off of our bank, come to some conclusions that we can test out in the models as well.
Hi there. I think that we would be happy to work with you if you're interested in proteomics, phosphorylation arrays, to do some multiplex ELISA-based technologies and really characterize your tumor of interest.
Thank you, Grace. The next question is for you. For CMET in the SRG model, what is the observation in the TME? Since the SRG mouse had no BT and NK cells.
So we have done some pretty extensive breast cancer tumor microenvironment work, and I'll be happy to send data to the person who asked the question.
Thanks, Grace. Why do you think the SRG OncoRat has been more successful in some cases for tumor studies when compared to mice? Have gene expression profiles in both tumor models been compared and then related to human tumors?
I think I'm gonna let Fallon take that question. Fallon, does your mic work?
Yep, I'm here.
Okay.
Yeah, hi, I'm Fallon. I'm also from Hera BioLabs. So this is a really good question, and one that we've fielded many times actually, about why tumor growth is seemingly better or faster in the rat. And the short answer is that we do think it's related to tumor microenvironment. With the PDX models that you see here, we actually implant the exact same size of tissue directly from the patient in both mice and rats. So it's not that we're putting in more tissue, and the rat seems to support it better. In both the PDX implants as well as commercially available cell lines, we see the same trends. Even if we put in the same number of cells, or sometimes we can put in less cells in the rat and we do get better growth. And we think it all comes back to tumor microenvironment. So as Grace had alluded, we've been doing some pretty stringent characterization of the tumor microenvironment, comparing growth in the mice and the rats. And it does look like the rat has much better stromal support and vascular support and early vascularization of the tumor as it is established. And we think all of that sort of is what promotes early growth and survival of the cells upon implant. If you're talking about differences in the mice and rats from a PDX standpoint, we've done genomics and RNA-seq at multiple passages in both the mouse and the rat, and we really don't see a lot of differences when looking back to the patient. So the tumor that we're seeing in the mouse and the rat is all the same with regard to, you know, how that patient tumor, the genomics of the patient tumor, if that makes sense.
Great, thank you, Fallon. You could probably answer this next one too. How do you measure tumor volume at Hera?
So currently it's in 2 dimensions. So a lot of our tumor modeling is subcutaneous. And so we measure with digital calipers, length and width, and then we use a standard published calculation of length times width squared, all divided by 2, and we report in millimeters cubed.
Excellent, thank you so much. In both presentations, examples of the establishment of mouse models that serve to guide clinical decisions were shown. I'd like to know what the rate is of success you had per indication and if it's realistic.
Hi, this is Jonathan from Certis. I can take that first one. It obviously depends on what your definition of success is. So we'll just start with, let's say, establishment. Our major body of evidence is in the work of sarcoma. And as I mentioned, despite the advancements of in silico data, many times those answers don't, or those results don't give an answer to sarcoma patients. So we published in JCO Precision Oncology that our establishment rate for high-grade untreated soft tissue sarcomas was around 65% or so, I can't remember. If you break that into subtype, such as leiomyosarcomas, liposarcomas, undifferentiated sarcomas, those increase. Undifferentiated sarcomas are about 89% success rate, successful establishment. So that's the first step. For success on the patient side, we're building that body of evidence. You know, I showed the case example for the pediatric rhabdomyosarcoma patient. We have more examples of that. Again, it's a matter of what your definition of success is. We find drug combinations that work well in the avatars, and when administered to the patients, it changes based off of where they are in their disease progression. But right now, we've been successful in that some of these patients have, are still alive and their tumors are controlled. I mean, does it cure them? Does the tumors disappear magically? No, that's not the case. But had they not had this option 2 years ago, they would probably not be around right now. So that's kind of how we define success. Again, we're building that evidence to make it more robust.
Thanks, Jonathan. I think patient outcome is a wonderful measure of success. Grace or Fallon, did you want to comment on that question at all?
Sure, I can take this. And the answer, I agree with Jonathan. It really depends on the type of cancer in the patient and where they are in treatment. For us, the majority of the tumor types that we've worked with, just based on our collaborators at the University of Kentucky and their expertise, has been non-small cell lung cancer. And I would say that, with what we've worked with so far, we have been pretty successful in establishing tumors early and using genomically guided treatment for them, and it's worked pretty well. But I can't speak for other cancer types because that's really been right now our main focus. But we are working more with prostate and ovarian cancers right now, so hopefully we can have an indication of how it works for other cancer types in the next couple of months.
Thank you. And the next question, Jonathan, you touched on this briefly, but are there any good in silico platforms for understanding the synergistic effects of drugs? And what should the approach be for in silico study of precision cancer medicine?
Yeah, this is a very good question. I'll start with the second one answering it. I think, it's when we started this company, a lot of people were saying, well, you've got this as a competitor, Tempest and all this other, and Foundation One, they're all competitors. And we don't view them as that. We think more information is better. We're in the information business. I mean, we're in the information age. We think of it as, an arrow in the quiver. And as I mentioned, some cases, the in silico, in most cases, I should say, in silico data is very impactful. It tells you how to narrow it down, what drug to try, and everything like that. But at the end of the day, it's still just a static representation of the tumor, and you don't have 100% confidence in that drug or the drug combination will work. There's also the technical issues of of heterogeneity. What did you sequence? Are all those cells expressing those mutations? Probably not. So which ones are the important ones like that? So again, it's a great starting point. It helps narrow down the decision. And we think all three panels of in silico, in vitro, and in vivo are important. And it's part of some of our pursuits as well in the next year to develop more in vitro platforms for precision oncology. To the first question about synergistic drug, we've worked with some companies who provide this. I don't, as far as the actual, let me look at the question again. Understanding synergistic effects, to my limited knowledge on these platforms that we've worked with, they don't quite say like how a drug would work or why or something, but there are a number of companies that suggest based on the mutations that double or triple combinations with these type of drugs would be more efficacious. And they have an algorithm and they rank them on, which chemo plus SMI plus whatever would be the best. I don't know the details of that. I can say that we've done this for patients who've gone through that and tried many of the suggested combinations. Some work, some don't, again, in the model itself. So again, I think the more information you gather, the oncologist has to take all the information, stand back, and then pick and choose and see. Once you can filter out what's, I hate to call it noise, but you take the results from all different platforms and then you kind of narrow down what your strategy is.
Thanks, Jonathan. That was a great answer. And I really like considering cancer to be your actual opponent here rather than more information or other platforms. Fallon or Grace, did you want to respond to that?
So we don't do that much in silico testing, but we do offer in vitro testing in commercially available cell lines, as well as cell lines developed from patient tumors prior to running efficacy studies in vivo. So we can certainly take cells from the patient and look at how they behave in vitro.
Excellent, thank you. So I think we all generally consider things that are customized or bespoke to be more expensive and to have very long timelines. So what can we do to reduce the timelines and the costs for this type of work?
So I can go first on this one. One thing you can do is when you first start your PDX, you start it in an SRG rat so that you get more tissue faster and you can take it then into mice or continue to passage it in rats and get to efficacy testing for your drugs quickly.
Yeah, I'll just piggyback off of that. It's a great response. And I actually reached out to Hera to see if we could work together to do that for our patients. Unfortunately, I got shot down by a regulatory in our ALAC committee on bringing rats into our facility. So we can't do that. What we are you know, it is a lengthy process, I think, and that's where it comes into all three approaches, is that in silico, in vitro is fast, in vivo is slower but more precise. So if you can incorporate all of them, then you can start delivering answers sooner. It might not be, you know, the end all be all answers, but you work towards that. And you know, also just in general, we Certis, we're continuing to research on ways to speed up engraftment, whether it be through certain type of gels, different implantation techniques. I can say that one thing is that we don't for our tumors, we don't always put it subcutaneously. Again, orthotopically, but we have some surgical tricks that we have to speed up and increase engraftment rates as well, and we're continuing to push that. If I may, I do have a question actually for Hera, for Fallon or Grace, with respect to initiating tumors in the rat. Obviously, you've shown great growth kinetics in that. Do those growth kinetics are they retained in the mouse once you passage it to the mice? Because I know you've done that. I'm just curious if you continue to get fast growing tumors, like if the rat has kind of primed those tumor cells in a way.
So I can take this. So the short answer is not always. So if we implant first into the rat and then put it into another rat, you're correct in that it is primed to grow well. And we often see a little bit faster growth at subsequent passages. And genomically and by RNA-seq, everything looks normal. So it doesn't look like we've picked up a lot of aberrations that would make it grow faster. And we typically don't go past passage 3 in the rat, just because we get so much tissue that we don't need to. And we can use P0 can be our working bank that we could just expand from there for efficacy. If we take a tumor grown in the rat and then put it in the mouse, we don't always get faster growth kinetics, but we get better take rate. So if we put a patient tumor in both the mouse and the rat, and it doesn't grow in the mouse, but it grows in the rat, we can then cross species transplant from the rat to the mouse and get a better take rate, but not necessarily as fast as it is seen in the mouse. And so we've actually used this to our advantage and to a lot of our clients' advantages, because they come to us with PDX models that grow horribly in the mouse, or a cell line that they really need to grow in the mouse and it doesn't really work well, but they really want to do their efficacy in the mouse. And so what we can do is we can establish tumors in the rat and then cut them up and put them back into the mouse and then run an efficacy with them. Whereas if they just implanted in the mouse, they'd either never reach efficacy or they'd be 3 to 4 months out before they could even run efficacy, if that makes sense. Did that answer your question?
That does. Thank you very much.
That was great. Thank you. I think panelist questions for other panelists are amazing. We have one more from an attendee. They said, I'm going to enroll for my PhD soon. Is there anything that is a must for precision medicine research?
It's a really open-ended question.
Perhaps they can reach out to you via email after
right. I'd say just stay on top of the latest news that's coming out for that.
Thank you. And
yeah. I also say don't shy away from immunology. Usually when I got my PhD, that some seemed a little overwhelming, and now everything's about immunology. So
I would agree with that. Don't shy away from immunology, but also I just from a personal standpoint, my PhD was not in oncology. I came to Hera, and that was just sort of the trajectory we took with the rat. And so I would say that, you know, even if you don't end up studying oncology in your PhD, it doesn't limit your potential out of your PhD. It's more about the skills you acquire, and both from a technical and from a cognitive standpoint.
Great, thank you. And just one final question to wrap things up. What are you hoping to see in personalized medicine in the next few years? And are there any big goals that you'd like to see accomplished?
I can take this one, or I can start it. So I think that that computer modeling and AI can inform in vivo work. And then I think that in turn, in vivo work can continue to inform modeling, so that perhaps decades from now, we won't need animals as patient avatars, because we'll be able to do detailed genotyping and proteomics work and then know very quickly what drugs to give to patients. But in the meantime, we need to keep using animals for PDXs so that we can learn more about how these tumors behave.
Yeah, from Certis' perspective, I can say that even greater than that, we need an overhaul on the paradigm and the dogmatic way healthcare, and especially oncology, is performed. We get a lot of patients who are kind of at their last end, and they don't know what to do because they've followed the standard of care, which didn't work. Then they went on drug 2, which didn't work. Then they went on drug 3, which didn't work, and now they come to Certis. So personally, I think just overall, the concept of precision medicine, personalized oncology, needs to shift and be applied earlier at diagnosis, so that patients understand that models can be made from their tissues earlier in the game than not. So our ideal situation here is a patient, they get diagnosed, they can get another biopsy that's sent to create a model, and then they go on their standard of care. Meanwhile, we're developing models to figure out what are the other options for the tumor. And should standard of care fail, then they can turn to the results of the model, rather than standard of care fails, and then they create a model, which takes time, and then doing that. Another thing too is just there's a hackathon by Brad Powers on precision oncology. And there's a phrase they use is tissue is the issue. And that's always the case. One, you have to fight these institutions to get the tissue out of there. Many times when it's a clinical trial or a large institution, that tissue gets signed over to the institution, and therefore the patient has no longer rights to their own tissue. So it's very difficult to get that out of the OR past the pathologist. So I think overall what our hope and vision is to bring precision medicine upfront towards kind of the earlier in the process, so that way patients have options with their own tissue to create models for them.
Wonderful. Thank you both. And thank you all so much for joining today's presentation. If you have any additional questions or you'd like to get in touch with our team at scientist.com or with any of the presenters, that information is available on this slide. And the recording of the presentation will be sent to your email within the next day. Thank you to our speakers for their involvement in the presentation, and thanks again to you for joining us, and we hope you found this presentation helpful.